proceed then with our last speaker, Professor Yuri Volkov, who's going to talk about nanomaterial induced protein citrullination.
It's forward, yeah. Good afternoon. I'm happy to give this talk here today because it's fresh. It hasn't been presented actually at the international conference of the high level, never because there was not sufficient information to justify it. So it's something fresh, and I hope you'll enjoy it. So first, today we're going to talk about this the effect of the citrullination which is induced by nanoparticles in human cells. And just for those who are not familiar with this process, I just remind that it's citrulline. It's the derivative of arginine, and it's not directly encoded by DNA, and it happens through the process of the post-translational modification, or so-called deamination of the arginine by the enzymes of the family protein arginine deaminases, or PAD enzymes group. And as a result, basic arginine becomes neutrally charged, and therefore there are more chances for the wrong folding of the proteins, so things go wrong if you get citrulline instead of the arginine. So, and mammalian cells have in general 5 types of those PAD enzymes, and they all have different tissue distribution. Of interest, those PADs are activated during apoptosis, autophagy, and so-called netosis, so neutrophilic cellular trap formation process. And citrulination is also involved in the process of functional regulation of histones. As you know, they are the gatekeepers of the cell cycle. And for example, the PAD4 inhibition, PAD4 can inhibit transcription because if it converts the arginine to citrulline on the histone protein, it blocks its methylation and DNA binding. So that's, you just abbreviate the process of transcription. Then very important is those PAD enzymes that rely on the high influx of calcium into cells, about 100 times more compared to the normal levels. And so therefore, it's usually associated with the processes of cell unhappiness, like cell death or just terminal differentiation on skin cells, for example, where PAD1 and 3 enzymes are participating. Interesting, those PAD2 and 4 enzymes both present in the joints and in central nervous system. So that's enough about the citrulination per se, otherwise I won't have time for the major things. But I'll give you the example of a couple of publications which appeared just nearly simultaneously with the interval about a couple of years in the 2004-2006. And one of those was in relation to the potential link of PAD rheumatoid arthritis, and another one was linking the smoking as a potential factor for the rheumatoid arthritis development. And one of the first author, Lars Klarskog from Karolinska University, he came to Trinity, to Dublin, and he was giving a talk about arthritis, and I never dealt with arthritis in my life, but he told us a very interesting story about the case history of Swedish postman, and I'll tell you a few words about this. That might be of interest for you. So, Swedish postmen, so what's wrong with them? So you see, in the old times, it was very romantic. I mean, the letters delivered across the water and by waterways was just, it's an engaged element of romanticism, and it was very pleasant. But as the times were developing and were going by, so the post was delivered like this, and the postmen are usually positioned in this compartment, at least in Sweden, for some reason, just behind the steam engine. Look at the hands of this postman. This is Van Gogh's picture. And this is the realistic picture of the rheumatoid arthritis. And what's so interesting is that once the postmen were relocated to the compartment, which was at the end of the train, the incidence of the rheumatoid arthritis in those poor postmen was dropped 10 times. So it was something that was relevant to the sitting behind the steam locomotive. And so when Lars told this during his lecture, I said, look, do you think it's just smoke or it's the nanoparticles? He said, I have no idea. So, but he said, the question is very interesting. So we decided next morning we dived into that story and decided to find the truth about this. And so a few facts we managed to discover is that this is cells exposed to silica nanoparticles, and green is just the amount of citrullination in the cells, and this is the activation of PAD enzyme, and as you see here, just it has the same control effect as the exposure to nanoparticles. We tried the whole panel, 2 cell types on the panel. We've got silica, we've got carbon nanotubes, single-walled and ultrafine carbon black, and as you can see here from the range, nearly all of this, but to different extent, all nanoparticles induced protein citrullination. So that was shocking, actually. And then we decided to check all this data, and it was true because the PAD activity was raised in both cell types, and then also the inhibition of PAD inhibition, it prevented the citrullination in the cells, and this is, you see, that's significant drop down in citrullination when PAD is not working anymore. And also the calcium levels also were sensitive, and once you use the verapamil to block the calcium levels, again the citrullination was blocking down. Finally, we did the immunoprecipitation and synthesized the proteins which were could be citrulinated, and it so happens that most of them, they are from a group of the cytokeratins, which was not a surprise, and also the plectin proteins as well. So that was seemingly okay in terms of in vitro characterization, but also the poor mice were inhaling carbon nanotubes for 6 months in NIOSH in Morgantown, Virginia, in the laboratory of our American colleagues. And as you see here, this is the area, and the green arrows show the citrullination in the lung tissue of those mice. So, and the long-term exposure causes citrullination in the animals as well. So, well, it was exciting. It was just a bit worrying as well for lots of people worldwide. So it got published in Nanomedicine London and got wide publicity. But after that, the papers were appearing. You see here, they always question mark. Dust is a trigger of arthritis, citrullination. Well, it's been spotted by our colleagues in Netherlands, and they actually posed nearly the same question as we did ourselves. It was very pleasant to see it. And also, as you see here, question marks. So lots of question marks were remaining, so we had to pursue this further to get them answered. And the questions were, to what extent the physicochemical properties of nanomaterials will affect the citrullination process? In other words, will all of them do this, or to what extent it depends on the shape, size, aspect ratio, and so forth. Again, it was inhalation model in mice, but what about other routes if you inject them? So autophagy, is it involved somehow, just long-term effects of the persistence of nanoparticles? Will those anti-citrulline antibodies be in fact induced in the animals? And is there any organ specificity in the citrullination phenomenon or not? So all these questions were remained there, and so as the time was going by, I said by today we managed to answer a few of those, and if not most of them. So first, it's the we used the carbon nanotubes in order to see the to what extent surface modification affects the process. With the same carbon nanotubes, single carbon nanotubes, but different modifications. There were covalent modifications. There were also the covered with BSA, and they were in combination with the covalent modification and malignant chemistry as well. So a few chemical modifications on the surface, and also in 2 different cell types. It's in the phagocytic and epithelial cells, and the color reflects actually, well, this is when everything is good, and red when everything is bad. So there's practically nothing is green here, you see, in this color maps. So what you can see here is that the irrespective of modification, if it's nanotube of certain aspect ratio, it will induce citrullination, but you can modify this. So you can play around this, which is good news. Aspect ratio, again, once I mentioned this, we decided to use totally different system. Okay, carbon is carbon, all right, but if carbon is, it's not only the carbon culprit, but it should be something else. So we used nickel nanowires and used them of 2 different lengths of 4 and 24 nanometers, respectively, calling them 4 and 20 short and long nanowires. And interestingly is that citrullination develops significantly faster with the with the long nanowires, as you see here, just it's over a matter of in a few hours. And whereas the short nanowires, the process is a bit delayed. So, but anyway, it's still there. So probably the short-term effect could be rather depending on the fact that it's frustrated endocytosis by the cells, and they are not happy straight away. So that's why we see the effects immediately. Whereas the short wires probably do this at a maybe later stage, autophagy dependent process. Also, we detected the formation of the autoantibodies in urine tissue, in urine sera. It was indeed the fact, and those were the nickel nanoparticles, just as an additional control. That's nanoparticles themselves here, and short and long nanowires. And this is the staining of the esophageal tissue, which was kind of treated with the sera from the mouse exposed to nanomaterials, and then citrullinated proteins detected, citrullination detected in the serum. So antibodies are there. So it's a process which develops in vivo as well. We looked at some of the organs in there, and these are the lymph nodes, and brown color shows you the degree of citrullination in that organ as well. And it is different. You see here again, long nanowires for some reason. By 2 weeks, their degree of citrullation is already lower, whereas the short nanowires, they continue acting up. We also checked the protein arginine deaminase activity in different organs, and again, short nanowires were the champions here, and it's been sustained over 2 weeks. So something keeps on going in the tissues of mice after that for a longer period. Well, finally, the moment of truth, we just got the samples of the mesothelioma, benign and malignant mesothelioma from the patients, mesothelioma of pleural mesothelioma and myocardial as well mesothelioma. And in this case, it's absolutely clear. This is the line which shows you the samples from those people with mesothelioma who had previously exposure to asbestos. So if there is exposure to nanomaterials, then you're likely to have your mesothelioma is related to the increased citrullination phenomenon. So coming back to the end, the most recent findings was a new twist kind of on smoking. We imitated the smoking cigarette in the lab, and we had the cadmium as a chloride oxides, and also we used the combustion. We just burn them at very high temperatures together with the ultrafine carbon, and then a related effect on the human cells. And as you can see here, this is a negative control. This is cadmium oxide, and this is ultrafine carbon black. So there is a significant induction of the citrullination levels, and what's more, I mentioned to you that the citrullination interferes with the function of histones, and some of those cytokeratins which are could be positioned in here in the even in the late in the cell nucleoli. Oh, sorry, wrong button. Okay, and most recent, it just arrived this month, the data from the Royal Cornwall Hospital from the United Kingdom, showing that there is a clear link between the rheumatoid arthritis and chronic obstructive pulmonary disorders. And as you can see here, from the 726 patients and 56 deaths, most of them were related to the respiratory diseases, COPD, smoking, or exposure to the dust, vapor, and gas. So 5 seconds. So citrullination, it's by now, it's known that it's associated with a huge number of the diseases, whether arthritis or not, including multiple sclerosis, COPD, IBD, and so this list is expanding. Mechanisms are not clearly known. It could be the ROS formation, could be netosis, citrullination of the particular proteins with the lack of neutralization of the lipopolysaccharide and others. And a growing amount of evidence really indicates that the environmental environmental and engineered, meaning that engineered also for nanomedical use, nanoparticles, could potentially serve as the factors directly contributing to the pathogenesis of this disease. So this is it. Thank you very much.
Well,
Thanks.
thank you very much. We'll take one question, although time is up.
Okay. Everyone is looking for coffee.
A question from anyone at the end?
Is it kind of a body's defense response to nanomaterial attack, or body's failed response to clear or take care of the nanomaterials, the attack?
Even PAD enzymes, yeah?
Yes.
I think it could be both processes. I can't exclude any, because as you see here, the dynamics of citrulination is different with the different aspects nanowires. So I presume that in one case it could be the response to the direct cell damage by some of the nanomaterials, and in other situations it would be, if it's extended process with autophagy, it could be response to the chronic process. Rather would be it would be the attempt to clear the damage which has been done to the tissue. So I think in parallel.
Okay, thank you very much, and thanks to all the speakers of the session. This was our last talk.